Yo, it's Dr. James Utley, your resident codebreaker data scraper, and I'm here to show you Nucleo. And if you don't know what Nucleo is, it's an agentic scientific research assistant that I built for myself. It kind of maps to a bunch of different models. It has the ability to search the web and the ability to search Google Scholar. And so we'll show all that and all the new features. There's been a lot of new features, and the outputs are looking even more beautiful and easier to read for the world. So let's get into it. The first one is let's just ask it a good question. Let's enhance the prompt because we're all lazy prompters. We all suck at it. So let's say, tell me about nuclear architecture in the lymphocytes. And I suck at spelling, always have, always will, but that's good. All right, now we're going to fire it off key command, which I'll tell you later. But it gives you a nice, beautiful prompt. So it enhances your prompt here, and you either use the original or you can send the enhanced prompt. Let's just send the enhanced prompt because we did it. Now, I don't use enhanced prompt enhancement every time because sometimes it's like it just gets kind of unnecessary. You should have the ability for prompt engineering, but usually like meaning steps in the way that you like to prompt to get the output of the model. But as you can see, it gives you kind of a great, beautiful output here. So we got chromatin organization, nucleoli, PML bodies, lamellar bodies or lamellins, and some discussion around gene expression and DNA replication and repair, the different T lymphocyte development, activation, and differentiation, its purpose in the immune system and cancer, and that's kind of what we get now. So we'll go ahead and shut that guy off. We'll hit speed research mode here, and what we will do is we will send. So let's just pick one aspect of this, right? Let's say, so can you tell me more about, let's see, T lympho differentiation. All right, so but now we're throwing it into speed research mode, which will give you a more kind of concise output and also search the web. So and I'll do some web search. Now, as you see, the output is more concise, meaning like it's just going to tell you like what it is, no fluff, um, and then it's going to give you the resources here. So you can see the bubbles that are in blue, and it just goes through it like where it got all its sources. So I call it references, but it's more like sources, and then you can go and you can check it yourself. Now, this is a function of it and not a bug, or not. This is a feature, not a bug. And what I mean by that is I didn't give you the ability to like output this in a format and then just copy and paste it because this is a co-intelligence agent. It's meant for you to enhance yourself, not for the AI to do everything for you, right? It's about the way of learning, right? So it's priming your mind with the different fonting and the texting, and then on top of it is when it's piqued and primed your curiosity, it's giving you the resources to go to, and then you can dig deeper. It's only supposed to enhance the human, not in place of the human. All right. So, but with that said, is you can see that uh, we have this monitor here, which is a takes and under it tells you what model you're using, and so in this case it's Gemini 2.5 Flash. I like the Google Gemini models, and that's real. I've already optimized Nucleo to work with Google Gemini, and it also because they have large context windows, and so you can see that the total tokens in the context window is there, which is right above here, and then you can also see how much usage you are in relation to the context window. So it's just something to keep in mind. And so when your conversations get too long, right, you can understand how many tokens you've used, and then reset the conversation. All right. So we looked at this. We have a web search capability in our speed research mode. We also have a web search capability. Now let's just take this part right here, and we're going to throw it in deep research mode. We're going to send it there. And the key command is Control Shift Enter. That just sends it instead of using the mouse. Okay, so this will bring it to this thing called deep reasoning mode. So it's sending it to a thinking model, which is Google Gemini thinking model. And then if you see right here, so it says the model you're using right now is Flash thinking. It says it right there. And then it goes through and it gives you the output. Now, what essentially has happened here is that it was really nothing to think about because it wasn't a question, it was more like a statement. But it did give you a snapshot of more depth on your research, on your references to dig deeper. So let's just go ahead and say, can you explain this to me like I am in fifth grade and need to teach others about it. Okay, and then what we're going to do is we're going to paste that in there and we're going to send it off. So it's going to send off that part back to the deep reasoning agent. It's thinking, thinking, thinking. I used to stream the response, and then it just got too messy. And I only do things that are like efficient for me. And I was at first I was looking at the streaming of the response, and then it just became like ridiculous, just unnecessary garbage. So I just wrapped it in that kind of thinking graphic, if you will. Okay, so it goes through that, and it'll tell you how to explain it as we just asked, and then it gives you the references just like that. All right, so with that, let's go ahead and we can you can see where you're at in terms of the token usage, which is right there. All right, now this is going to clear the memory and kind of clear the whole chat. So, and I put it here on the input bar just because it's like more efficient to use instead of scrolling all the way up to this trash can, which does the same thing. But if you're like, you just kind of get too far down the line and you're like, okay, fuck it. Like, let's just change it, get back. All right, so there we are. All right, let's start over with, can you tell me about functions of neutrophils? All right, the beautiful immune system. All right, we're going to enhance the prompt. Of course, I like to do that when I start. Okay, and let's see. Are we going to base model it? Yeah, we'll base model it. So we're going to hit, we're going to we're going to send the enhanced prompt to the base model, and we're going to wait for it to return a beautiful output that we can all enjoy. All right, so, and if you see that it goes over the function of a neutrophil and everything that you just need to know, and it'll drop it out now, or drop it in the chat. Now, I want to highlight this part right here, which is kind of like one of the undervalued resources of Nucleo, is that it has a RAG database, which we call the brain bank. And the brain bank, so it searches the RAG database. So this is all curated research that either myself or those that I trust to then provide research to the database. And it's all in the domains where we want, the domains in which I have like higher acumen, not expert level. I just hate that. I hate calling myself an expert because I'm like always learning every day. But like higher acumen than I would say most, which is, of course, like cell biology, immunology, genetic engineering, genetics, these things, stem cell research and science, which is all a lot related, data science, machine learning, these type of things. And so, like, those are the domains in which curated research is, and we're just building this database every day. And with that, building the ability to like look through the database, which means index it correctly to find the most valuable outputs. But I would say that this knowledge source bar will only get better because there'll be more context. Right now, this number is actually wrong. We have probably about 100 more papers that need to go in, but they just need to be verified before we update the brain bank metric. But the standard rule of thumb at this point is if it's 14%, which means if it's double digit percent, then you're getting, in contrast with the web search capabilities, you're getting a pretty accurate answer. So that's one. Now, I want to also highlight this, what we call short-term memory, which is a little floppy disk here or a little hard drive disk, whatever you want to call it these days. And what it does is it continues to pass the context from prior conversation into the next one, right? So that is a valuable tool at some point, but you have to understand that LLMs or generative AI can be very limited by hallucinations. And if you're using web search, you're passing the context, you have a RAG database, you kind of have these like 3 points of verifier that'll keep your conversation on the right track. Now, it can get out of hand if you're not careful. So what I'm gonna show you what this means. I'm gonna say, can you please summarize the next prior conversation, let's say in 100 characters or less, or 100 words. That's not characters in 100 words. All right. So if you see it passes the whole context window of what you were talking about, and it's a useful tool. So right here it says the conversation has began with a detailed view of nuclear architecture and T lymphocytes, covering chromatin organization and all of the things that we talked about. The discussion then focuses on T lymphocyte differentiation, explaining how naive T cells differentiate, and we sent that to the base model. So let's go. Well, actually, let's take that because it threw this note here that says note Google Scholar search was unavailable for this query. Possible reason is the conversation context may be too large for Google Scholar, and it gives you a pro tip, which is try to find one topic and then ask it directly for the research, which is actually a really amazing feature, which I'll show you right now. Which is the one that I'm very. So let's just take this one, which is nuclear architecture and T lymphocytes, is what we kind of started with. Let's shut this guy off. Let's clear the chat. And now we're back here. Now we're back here at the top, right? And we're on the base model and we're all cleared out. Tokens are 0, etc. All right, now let's say, can you give me the latest research on X, which X in this case is nuclear architecture in lymphocytes. So what we're going to do is we're going to send that, and this is in the base model, and what it does is it does a search, and it's going to give you what it looks like is one study. Now, it tries to give you between 1 and 5 studies via the Google Scholar, you'll see that it's, you can click in here. It says like when it gives you a little summary of how old it is, and then kind of a brief summary there, right? So let's try it again and see if we can get a little better output. Can you give me the latest research on stem cell therapy with MSCs? All right. So, and we'll see what happens here. So it's going to hit Google Scholar and see that this time it's going to give you the overview with a citation, so you know where it's coming from. And then when you hit the references, it gives you 3 studies to take a look at. And it's not like a whack-a-mole, meaning like you're not going to get different studies every time. It's going to have reproducibility, but the point is that when you're doing a literature review, it's getting in the right way because it has the context, and then that context is reinforced by published research. And let's click on one of them so you know what I'm talking about. And then it goes directly into the paper. You should be able to download the paper or to view the PDF, like in this one here, which is actually cool. Axon regeneration, rat sciatic nerve defect model. So that's cool. So let's take it back. Let's take it back and look at this. And okay, so it's hepatocellular carcinogenesis, nerve regeneration, which was number 2, which is what we looked at. So it's really an aid in helping you research. Instead of like just trudging through the depths of like PubMed or Google Scholar or wherever you're looking, it's a bit more interactive. I enjoy it. I learn it. I've been like actually surprised on some of the papers on the output because I've like learned quite a bit. Okay, so things that you won't see is if you see here, there's no knowledge bar. So when it's searching Google Scholar, it's generating model and Google Scholar. It's not looking at the RAG database, which is okay. I think that's the most efficient. The Google Scholar or parsing out these papers is a bit fragile in some way, which means that if Google Scholar doesn't really get the query, or if the query is too large and the model can't API call to Google Scholar, then it'll just error and say that it's too big, and you just have to adjust it. So if you're talking about like kind of the workflow, all right. Oh, the last thing before I talk about workflow is like this is orange. So orange is studies, blue is web search. So you see the 2. So if you get an orange bubble, like 1, 2, 3 here, then you're getting directed to a study. If you're getting a blue bubble, then you're okay. So let me just show you. Then you're going into a web search, right? So can you tell me about eosinophils? All right, so let's just do that. Let's just throw it out there so you know, so I can show you the contrast so you know what I'm talking about in terms of the design feature. All right, you see here, It gives you speed research, so it's giving you the down and dirty and nothing more than you need to know about what an eosinophil is, and that's true. And then if you see here, these are all blue. These are blue bubbles, which means that they're searching the web. That doesn't mean that they're not searching a paper on the web, but it's a web search. And it says right here, web search. And if you look right here, it shows you how many times the paper is cited, where it came from, et cetera, and that type of stuff. So it's better to and if you see that we hit that off on the speed research, and you'll see on the output here, it's nothing, down and dirty, which means it's everything you need to know. But it also hits the RAG database, and you see that we're greater than 10%, so we're probably doing pretty good in terms of accuracy. And that is that. So let's go ahead and clear that guy out. And if you notice that on the sidebar here, you can kick off a new chat. That'll kick a new chat off. And you'll see that there's the context window and you got your, everything there. But if you clear the chat there, it should get you back to 0. This one is like a hard reset, kills everything. But if you wanted to like continue on your task and then you wanted just to add a new chat, you just hit this little green button. It'll keep the context window and it'll keep some of the kind of residual memory going on. And then if you want to pass the context, remember short-term memory is right here. And that will pass the context back into from conversation to conversation to conversation. All right, let's look at some cool sidebar UI stuff. And okay, you can become a pro member, which is amazing if you want to support the project. This is a community project. It will continue to be a community project, and I'll explain why. You can join our AI community, which is the Institute for Prompt Research. If you click there, you're going to see that we have a community with 48 members here that are like-minded AI people trying to make their way, trying to be a collective of grifters, grifting the AI world. Nah, I'm just kidding, but sometimes it feels like that with all the grifters, trying to build real tools. I guess that's all I'm trying to do, and I'm trying to build tools for myself, which hopefully could help other people. You can export your chat at any time, and then it exports it in a TXT file. So if you want to like throw it in Notion or Google Colab or whatever, it can do that. I would say that this is not like really a coding agent, if you will, even though I will say like, tell me about linear algebra and give me an example. So let's do that. Let's just throw it in deep research mode and see what we get for the output. And okay, so yeah, it's not like a coding agent. It will do some coding, but it's not designed for that. There's too many different coding. At first I had some coding aspects in here, meaning like I was a Python dev, so it was like had Python and it had some R capabilities, SQL, etc. But I've kind of at this point taken that out. And if you see that right here, so it gives you a beautiful example, and I love the output now. So it's like really about the design element, that it's like nothing more than, I just asked about linear algebra and it gives you these like beautiful, first we calculate the A and it kind of gives you the step by step on, and it can really level up your game. And then if you want to like dive more into it, you can just dive more into it. Like I'm really happy with the output. I love the colors. It really helps me learn. Like I can just like scan and then find where I want to go. So like this right here is beautiful. It's like vectors, vector space, and it kind of gives you this visual representation of this vector. And you can copy all these and then throw them where, you can put them in your papers or whatever. So it's kind of a beautiful output. And whoever's like listening, it is like 21 minutes into this video. So I really appreciate I really appreciate that. Okay, so you see, we use the RAG database there. So but let's talk about those last things, which is this is an upload research where those that have been ordained to the beauty of Nucleo and can upload to the database, like this is how they do it. Which if again, it's a community project, but it's not meant for it's meant for scientists and those that are curious about science, and to really help that as a tool. So you want people to be a user to then build the tool, right? And that's really the next focus, is uploading research to make sure that the brain bank can continue to. And of course, this is just me, So if you want to be part of a community project that is in the world of AI, and this is an open source. Like I even collaborate on the code, open source the code, not to anyone, but what I'm saying is to those that really give a shit, I will definitely do that. I just want to keep this thing going, and I'm trying to do everything I can to keep it going and keep building on it. And with that said, is like if you're participating in the community, sign the guest book. These are all the people that have participated in Nucleo, and sign the guest book, and we know that you're part of the crew here, and we're trying to build the most community-driven, scientific, intelligent, agentic workflow that exists. And I think I totally believe in the wisdom of crowds, and we can totally do it. So, anyways, with that said, is I'm going to sign out here now. Oh, and we'll see you in the future on all the next updates and the features. So it's found at nucleo.chat. Peace.